Hello and welcome to part 3 of the ESAM webinar series. My name is Alex Howard, and I'm a part of the SARDI research team that performs regular analysis of the national ESAM data to prepare monthly reports for red meat establishments, MLA, and the Department of Agriculture. In this video, I'll be going through some real establishment ESAM reports and highlighting some of the important things to look out for when reviewing your own ESAM reports. So this is going to be an example of a steer heifer establishment ESAM report, which is going to contain summaries for TVC, both carcass and carton, E. coli, carcass and carton, Coliform, carcass and carton, and also Salmonella just for the carcass. For the E. coli results, we're going to also look at some sheep or lamb report examples, as E. coli is more commonly detected in those species and it can be a bit easier to identify some working trends. So TVC is our quantitative measure of all types of bacteria. Now this bacteria comes from a range of sources, including our animal hides, plant equipment, hands, or the air. And a key thing to remember with TVC is we almost always get a count, leading to very high prevalence, north of 80% for every single one of our species. Now TVC is used as a general indicator of shelf life and poor processing hygiene. And in terms of species differences, you would expect your sheep and lamb to be typically around half to 1 log higher in your median counts when compared with beef samples. So taking a look at our TVC summary for this plant, we can see that this plant has had a high number of TVC detections, so really high prevalence, but this is no cause for concern. As we mentioned just before, TVC is our quantitative indicator, so we expect to get a lot of positive counts. And in actual fact, the more positive results we get, the greater indication we're going to have of our process hygiene. So looking at the counts themselves, they're pretty stable from January to February, as you can see. And then over the last 3 years, they're tracking pretty well, around 0.05 of a log below the national average, so pretty stable. And taking a look at the standard deviation, which is a measure of our variability, we can see that that's pretty consistent as well, which is all pointing to, good sign for this plant's process control. Now taking a look at the monthly TVC summary for this establishment, we can actually see that they're pretty consistent from month to month. There's a little bit of maybe seasonal variation going on here, December to March and then again here, but not a lot really. And performance overall is in line with national data in terms of the median, as we saw with our summary tables as well. There's no real alarming signs here. I mean, there was this odd month here that was a little bit higher, but it was corrected straight away. So looking pretty good for this establishment. Now taking a look at the weekly TVC summary. Now you got to remember here there are few accounts contributing to each one of these boxes. And this is why we see a little bit more variability coming through within the establishment plot. Seeing a lot of little short-term trends here. So within each month, the counts week to week tend to increase, sort of month to month. And over here, there was a bit of variation, but it's been brought back in line. That probably corresponded to that little peak in that monthly plot that we identified before. And interestingly to note again, in February here, each week, their median counts have increased sort of steadily. So something to keep an eye out at the end of the month. It looks as though there's a bit of a short-term trend where counts are increasing. Overall, again, pretty consistent. It's a lot more difficult to tell if there's a seasonal variation going on because there's only actually 1 year of data on these plots because they are weekly plots. So just got to keep that in mind. Again, taking a look at the national comparison for this month's TVC results. So these are all the Australian establishments combined on the one plot with their last month's TVC data. And our plant here is indicated by the grey shading in the box. And we can see that this plant has pretty middling performance. It's pretty much dead in line with the national average, and some solid variability indicated by the height of the box, not too high, but not very small. The plant on the left is recording some really low counts, and judging by the size of the box, we can see that they have actually very little variability. And this indicates they're having some really good process control going on there. And then on the right, we can see an example of a plant whose median count, while is in line with the national data, they've got a lot of variability within that past month. And that's actually letting them down a lot in terms of their high trending counts. So that's a bit of a sign, maybe some poor process, something's gone wrong there. So taking a look at the TV summary for this plant's carton results, we can see that their TVC prevalence is still fairly high. It's a bit down in terms of compared to their carcass data, but still, you know, above 70%, and certainly over the last 3 years, almost 90%, which is bang on what we would expect. In terms of the counts, we can see that they're trending a little bit above the national average by about half a log in terms of their last 3 years, but they've certainly brought it down within the last couple of months, but still running a bit higher than the national average. So as we can see from this figure, carton TVC tends to be around 1.5 log higher than our carcass results. And this is really expected, given if you think about carton meat, it often contains boneless trim, it's gone through a greater level of process compared to the carcass, and therefore has had more opportunity to pick up that bacterial contamination. It is interesting to note that for this plant, the median count and the variability of their carton TVCs actually appears to have come down over time. So this would indicate this plant has made some improvements regarding their process control of their carton meat, and is actually on the right track. It's also worth mentioning that these are in 2 different units. So your carcass is a swab sample, so it's surface area, CFU per centimetre squared, where your carton is a sampling, so it's CFU per gram. And that also contributes to the difference that you're going to see in terms of your carcass and your carton TVC. So it's an interesting comparison, but pretty expected that we would see carton TVC running a fair bit higher, around as we said, around 1.5 log higher than your carcass results. So E. coli and coliforms are 2 organisms that are indicators of fecal contamination. Now, they are not normally causes of illness, but their presence is used to indicate that other pathogenic organisms of fecal origin may be present. Therefore, they are important parameters to measure for our processing hygiene. Now, in terms of the prevalence and average counts that you would expect, it varies between species, with sheep and lamb tending to have a slightly higher, 10 to 15% higher prevalence, and a little bit higher concentration, around half a log higher concentration when compared to beef. But both of these organisms, E. coli and coliforms, are sporadically detected, and will have lower prevalence and much lower counts when compared with TVC, as we can see here in this table. Now, E. coli and coliform results tend to be very similar, given their closely overlapping biological relationship. So in the interest of time, what we're going to do in this presentation is actually focus on E. coli results mainly. We'll draw in some coliform figures here or there, but we're sort of going to combine the 2 because it's very similar when interpreting the results. You can come to the same conclusions. I suppose the other thing to mention is, again, with the low counts that you would expect with E. coli. So our log counts are actually going to venture into that negative range. And so just important that we consider what a negative count actually means. So the median count for cow bulls around negative 1.08, which corresponds to a raw count of 0.08, which is pretty much bang on our lower limit of detection. So we're talking about very low counts here. Now, this is our E. coli summary. Now, note the really low month-to-month prevalence. So remember, this is a steer heifer report, and beef processors tend to have fewer issues with generic E. coli and coliforms than sheep or lamb enterprises. The counts are also very, very low, which is a positive sign, but expected. And again, those negative log counts can be confusing, but just remember when the raw count drops below 1 colony forming unit per centimetre squared, they do appear as negatives. So in that February 2018 column, we have a median count of negative 1.097 logs colony forming units per centimetre squared, which translate to that 0.08 CFU per centimetre squared, which is right around that lower limit of detection for E. coli. So switching over to a sheep plant report, we can immediately see that higher prevalence coming through, whereas before it was around 3 to 5% mark. Here we've got 37 and 28% in the last 2 months in terms of prevalence. And over the last 3 years, this establishment is performing about on par with the rest of the nation. But those last 2 months actually have an increased number of positive detections, something to keep an eye on, just coming through. In terms of their counts, again, this establishment is pretty much in line with the rest of the nation in terms of their median counts. But we can see in February there was a small spike with monthly counts jumping to 0.107 log, which is around the 1.3 colony forming units. But if we look at the maximum for that month, just down here, it's only 1.38 logs, around 24 colony forming units. So they are not getting super large spike counts, just to put it in a bit of context. So this is a monthly prevalence plot. It tracks changes in E. coli prevalence from month to month. And we can see that this establishment tends to be up and down a bit, with seasonal peaks occurring around that June to September mark. And note that the same peaks actually are occurring in the national data as well, with the exception of 2017, where we might have had a bit of a flat spot here. Now it's important to put into context, this plant only conducts around 50 E. coli tests a month. So when we see a large change in prevalence like this from month to month, we have to understand that that may just be occurring because of 3 or 4 additional detections. So you just got to put it in a bit of context. And then again, if we look at the weekly, it's the same thing again. This is back down to 13 to 14 tests a week. So even just 1 or 2 additional detections a week is going to change your weekly prevalence drastically. So that's what's occurring here. Again, from a weekly plot where you've only got 1 year of data, it's very difficult to see seasonal trends. You really want to use those monthly plots to identify your seasonal trends. So this is a monthly count plot from this establishment and nationally. And we can see that this plant has a little bit of month-to-month variability going on here, but that's pretty normal for E. coli because it's infrequently detected. And the within month variability indicated by the sort of the height of the boxes is pretty good, with the exception of a few odd months out. And unfortunately, the most recent month here, February 2018, was a bit more variable than normal. And that's going to be reflected in the next plot as well. But comparing with the national data, pretty good, pretty much in line. So in this plot, as we said before, that it's a national comparison. So we have all of our different sheep establishments here and their previous month counts reflected in a box plot. And you can see this high variability has really kicked this plant this month, hit them pretty hard. Apart from that though, their median counts a little bit higher than the national average, but it's not too bad. But yes, again, that variability really comes through there. But then again, if they have a low variability month next month, then that will again be reflected in the national plot. So your fortunes can sort of change month to month. So taking a look at the E. coli time plot. So this plot shows positive E. coli results as red points, and it shows where they sit in terms of those defined limits, the little m and big M. So this has come from a sheep report. So those defined limits for little m are 5 colony forming units per centimetre squared, indicated by that little red dotted line down the bottom there. And the big M limit is 100 colony forming units per centimetre squared. So we can see that most of the positive counts occurring from this establishment are occurring in that acceptable range, so below little m, and also in the marginal range, so between big M and little m. We can see there was one unacceptable count that occurred in May 2017, but it appears to be an isolated incident, with the rest of the counts being pretty stable. And again, picking up on that latest month of data, we can see they had looks like 1, 2, 3, 4, 5 counts that occurred within that marginal range between the little m and big M. And that's probably what's contributed to that increased variability that we've seen over the past month. Now this plot also includes a national comparison, and you can see how many detections there are in between the limits and also above that have occurred nationally over this same time period. But really it's a bit of a mess. Not a lot you can draw from the national plot in terms of this. So just tend to focus on your own results here and how they're tracking, and that you're not getting lots of clusters that are above the defined limits. Now if we take a look at the same plot from a steer heifer report, we can see straight away that the limits are different. So the lower limit, little m, is 0 CFU per centimetre squared, and the big M limit is 20 CFU per centimetre squared. We can also see straight away that there are a lot less positive detections, which we would expect, given that this is beef. Now, most of the positives from this establishment are occurring again in that marginal range with no unacceptable counts. And this is showing that this establishment has a good track record of limiting the amount of fecal contamination that is occurring. Because sometimes when we get E. coli counts, they do tend to spike quite highly. So again, in the national data, the majority of counts occurring in the marginal range, but there are quite a few unacceptable counts. But again, it's pretty messy, so best to stick to just looking at your own establishment and how you're doing over time. Now, not everyone actually enters their results for E. coli testing of cartons, but the Coliform data is pretty good and pretty consistent, so we'll use that as an example here. Remember, this is from a sheep report. So if we take a look at prevalence, we can see that this establishment's got pretty consistent month-to-month prevalence, and it's been pretty much in line with data from the last 3 years from this establishment. But if we compare with the national data over the same time period, we can see they're running at quite a bit higher prevalence. Now, why is this occurring? Well, one potential answer could be to do with the proportion of their carton meat that is made up of trim. So if they have a high proportion of their carton meat that's made up of trim, potentially some of that contamination that has been removed from the carcass is contributing to a higher number of detections in their carton meat. Now, we've got to ask ourselves, is this a problem? And the only way to find that out is to look at the count data. So if we look at the counts for this establishment, we can see that they're actually running pretty well in line with the national data over the last 3 years at least. So while they may be getting a higher number of detections, it's not actually transferring over into a high number of really, really high counts. So it's not as big an issue as it might seem at first look. If we take a look at the comparison between now carcass and carton coliform data, we can see that carton coliform concentration tends to be around 1 to 1.5 logs higher than that of the carcass. And this is again really expected because carton is made partially up of trim, so some of that contamination removed from the carcass is contributing to increased counts on our carton. And also, just if you think about carton meat, it's gone through a greater level of process compared to carcass, so it's had a more of an opportunity to pick up that micro-contamination. And then lastly, there are 2 different sampling types. So carton meat is a sample versus carcass, which is a swab sample, which is much more sensitive and picks up much lower levels of micro-contamination. So all of those things contribute to this really broad difference that we see between our carcass and carton results. So moving through to Salmonella, which is a globally significant foodborne pathogen. It predominantly relates to poultry, sheep and pig products occasionally. Testing is required when exporting to the US. And in Australian red meat, it's rarely detected, as evidenced by the really low national prevalence figures we can see there for the last 3 years of ESAM data. So as we can see, our establishment here, has no issues and has had no Salmonella detections at this establishment over the last 3 years. So no serotype information is provided. And this is a pretty typical result. We can see only 6 out of over 10,000 tests nationally for beef. So pretty infrequently detected. This is an example of a table that would be included in your ESAM reports if you have returned previous positive detections for Salmonella. So it gives you the information regarding the test date and the specific serovar or serotype that is detected. So that information is just captured there. So that's going to wrap things up for this ESAM webinar. So next time you receive your monthly reports, it should really be straightforward. Hopefully this information is useful in helping you work through step by step your results and helping you develop some comments on your own level of process control and maybe what's occurring at a deeper level at your plant. For plants who export beef trim for grinding in the US, the next webinar is going to focus on interpreting the specific E. coli 0157 and STEC monitoring reports. So we'll discuss the importance of STEC testing in Australian beef. We'll break down a few examples of establishment E. coli 0157 and STEC reports. And we'll focus on interpreting some of the key results and also looking at some national trends. Hope you enjoyed this ESAM webinar. We'll see you in the next one.